Welcome to SGT Times Explainer. I am Ankita. Today we will discuss how our brain creates long-term memories and DNA can be broken and repaired. According to research published in Nature and done by experts at Albert Einstein College of Medicine, negative events induce DNA to break and repair, which aids in the formation of long-term memory. DNA damage in the brain, followed by repair, helps consolidate long-term memories according to a new American research of adult mice. The peer-reviewed study done by experts at the Albert Einstein College of Medicine in New York and published in the journal Nature on March 27 demonstrates that as long-term memory emerges, some neurons in the brain receive so powerful electric pulses that their DNA snaps and breaks. According to the study, this activates an immune system reaction which induces inflammation and eventually aids in the healing of the damage to solidify long-term memory. This is the first time DNA damage has been linked to long-term memory. Typically, breaks and damage to both strands of DNA are connected with a wide range of disorders in the body. However, DNA damage in the brain has previously been linked to learning. The study is also noteworthy on another level. Alzheimer's disease, an advanced type of dementia, is known to proceed as a result of protein accumulation in neurons. According to the latest discoveries, it is this DNA damage and repair machinery that malfunctions and causes mistakes to accumulate in persons suffering from neurodegenerative disorders. To determine the effect of known DNA damage on memory formation in the brain, the researchers taught adult mice to be terrified of a certain sort of environment. They gave the rats a tiny electric shock to induce a fear reaction when exposed to such surroundings. The scientists next looked at gene activity in the hippocampus, a brain region thought to be important for memory, and discovered that 4 days after the electric shocks, genes involved in the inflammation appeared in a group of neurons. However, 3 to 4 weeks later, the inflammatory genes were reduced in frequency, indicating that DNA strands had been repaired and the rats' long-term memories of electric shocks had been established. During the investigation, researchers from the Albert Einstein College of Medicine discovered a protein called TLR9, which they said was responsible for activating the immunological response to DNA fragments. However, because there is no infection, the immune cells are not required to fend off a foreign body. Instead, the ensuing mending and recovery processes occurs independently, repairing the DNA damage. When the researchers genetically modified mice to delete the TLR9 protein, they discovered that the animals had difficulty remembering their training and were less scared when exposed to the shock-generating environment, indicating that no long-term memory was formed. There was also one discovery that baffled experts. They found the role of a tiny cellular component called the centrosome in DNA repair. While centrosomes provide several cell functions, their primary job is to aid in cell division. However, because mature neurons are not known to undergo mitosis or division, researchers are perplexed about their function in DNA repair. The discovery has also raised further issues, many of which are related to the specifics of how DNA breaks occur and whether this process occurs outside of the hippocampus in other sections of the brain, according to researchers. That brings us to the end of today's SGT Times Explainer. Thanks for watching.